In section 5, we'll be looking at the presentation of antigens to T cells. Unlike B cells, whose secreted antibodies see whole antigens, T cells see very small peptides of between about 8 and 13 amino acids long, presented to them by a molecule called MHC. And MHC stands for Major Histocompatibility Complex, and we have MHC1 and MHC2. In humans, we sometimes call these HLA, which stands for human leukocyte antigen, because these MHC molecules were first found to be antigenic on the surface of leukocytes and important in tissue rejection during transplantation. HLA is hugely polymorphic genetically. There are many HLA proteins, and a number of them exhibit immense genetic polymorphism. So we'll talk about the genetics of MHC or HLA, and we'll use those terms interchangeably because all this discussion is in relation to humans. How do peptides from viruses or bacteria or other pathogens get presented to T cells? How do they get processed? How do they come into combination with these MHC molecules which sit on the surface of mammalian cells? And that's the subject of endogenous and exogenous antigen processing. MHC loading refers to adding these peptides onto the surface of MHC. We'll look at the structure of MHC in detail. This is just an introduction slide. Why should medics be very interested in HLA? Well, a number of reasons. Primarily or originally, we're interested in HLA because for tissue transplantation or organ transplantation, these HLA molecules are antigenic, and if we have a mismatch between donor and recipient, then we get rejection. And because there are so many different genetic variants, then this is a much more tricky subject than the AB or blood group antigens. So for the sake of transplantation, the genetics of HLA are very, very important. And also more and more people are appreciating the correlation between this genetic polymorphism, various variants, and medical conditions and susceptibility to infection. And here's an example of that from a recent research paper, looking across the whole of our genome, chromosome by chromosome, and counting the number of SNPs, which are single nucleotide polymorphisms. Each dot here represents one specific mutation, one specific SNP in the population as we look across the whole of each of these chromosomes. And what people can do is to look for an association, we call this a genome-wide association study, between these genetic polymorphisms and a specific trait. And if this association is found, and if the statistical p-value is low enough, then we may be have good reason to believe the possibility of a causal relationship between the two. And this is often plotted in something called a Manhattan plot, which is shown here. If any of you go on to actually read and study the medical genetic literature related to this subject, or actually related to any subject, if we're going to study the medical genetic literature, then you will very commonly come across plots like this one here, where the p-value is actually given as the negative log of the p-value. So as we get lower and lower p-values, because we're taking on the y-axis the negative log, we actually get higher numbers. And in relation to our HLA study, the association is absolutely remarkable. And when we come to chromosome 6, which is where the HLA genes are found, we find a whole number of polymorphisms. Each dot represents a specific polymorphisms, which is massively correlated to the thing being studied, which in this case, from this recent paper, is self-reported allergy to the antibiotic penicillin. So some people are highly susceptible, or some people are allergic to penicillin, and this is related to the presence of specific polymorphisms, several of them here. So most of the SNPs are not related to allergy to penicillin, but quite a number of them are, a big number are actually related to that. So a value of, say, we're getting close to 30 here, a value of 30 means that the p-value is 10 to the minus 30. So that's a very, very, very strong association. And you'll often see these type of plots called Manhattan plots in the medical genetic literature, where people are looking for genome-wide association between some particular SNP, single nucleotide polymorphism, and a trait of interest. You can find in the research literature many examples of plots like this showing correlation between various HLA genes and susceptibility to COVID-19, susceptibility to influenza, susceptibility to all viruses, or any particular virus. And many people misunderstand this. For example, two people apparently healthy, similar age, infected with COVID-19. One has a mild flu, the other one has severe acute respiratory distress and needs to be placed on a ventilator and is at risk of death. What's the difference between these two patients? And this may come down to genetic factors between these two patients. And one of the common factors of difference here is polymorphism, difference in these HLA molecules which are presenting peptides. Some polymorphisms have a much greater ability to present peptides from infecting COVID-19 than other ones, and that difference will show itself in a different prognosis, medical outcome for that infection. And what that means is that it's not really fair to say that someone has a weak immune system, and that's why one person is susceptible to very serious outcomes for, say, COVID-19. It may be some people are immunocompromised and they're across the board, their immune system is weak. But for most of the cases, it's actually that there's a kind of a balance in play here. Each one of us is has a very high capability of our HLA molecules to present very specific peptides, which means that we may have a very good capability to fight off COVID-19 infection. However, the polymorphisms in our HLA molecules may be very poor at presenting epitopes from another infection. So it's a game of chance from our perspective. Those of us who were able to fight off COVID-19 with very mild symptoms, that does not mean that we somehow have a super immune system. It just means that the particular alleles of MHC that we have are very good at presenting those peptides. When it comes to another infection, things may be reversed and we may be very poor at presenting that. Many autoimmune diseases as well are affected by HLA. Diabetes, for example, diabetes type 1 is related to haplotype groups called DR3 and DR4. And I'll talk about haplotypes when we get actually specifically to the genetics section. But here, I just want for those of you who are medical students, which is a great many of you taking this course, to appreciate how important the genetics of HLA are for a huge number of diseases. The presentation today is actually a very condensed presentation. I want to just highlight the main points and introduce the molecular pathways here with endogenous and exogenous processing in a fairly straightforward type of a way. So let's look now then at this structure. We've got a peptide here in orange from a pathogen, and in blue, 2 shades of blue, we have the MHC molecule. So let's look at that structure in the next slide. Okay, so we have MHC class 1 and MHC class 2, and for each of these I'm showing you a sideways view, and also in the corners here for MHC class 1 here a view from above. So this is a view looking down here, and the peptides are binding in what's called a peptide groove, and there's a peptide groove for MHC class 2 as well. That's for MHC class 1. And here is a peptide in MHC class 2 sitting in this groove. And this groove is made from, it's clearer here in this, MHC 1. It's made of a beta sheet, and it's these arrows lying underneath the peptide, and overlaying the beta sheet are these two long alpha helices, and within these and sitting upon this beta sheet the peptide sits. And you may notice a small difference between the peptide here and here. See, the structures of MHC do look very similar. Look at this loop here and this loop here, but then there's another loop here in MHC1 which is actually much bigger than the corresponding loop of the MHC2. We have a loop down here for MHC2 and likewise a loop here. So there's a considerable conservation between these structures, although there are some differences, and the peptide sits tightly in the groove for each. The peptides for MHC2 are a little bit longer, about 13, even up to 18 residues in length, while for MHC1 over here, the peptides are shorter, about 8 to 10 amino acids in length. And we'll look at the determinants of peptide binding in the next slide, but still on this slide, let's look now at the sideways view. So now we're looking sideways on, and the peptide is going into and out of the page here. For MHC class 1, there's a single MHC chain, which is the alpha chain, and it's divided into two domains, A1 and A2. The color coding here matches the color coding here. So we're actually looking at two domains of the same chain, which are associated with each other. Alpha 1, alpha 2, alpha 3. You can see there's some disulfide, intramolecular disulfide bonding helping to keep this molecule in its structure. And there is a transmembrane segment and a cytoplasmic tail. In addition, there's another domain which is basically a molecule called beta-2 microglobulin. And there are many MHC class 1 molecules. There are 3 of them which are called the classical MHC molecules, which we'll look at more when we see the genetics aspect. But there are 3 classical MHC class 1 molecules. We have 3 genes for MHC class 1. Each is a little bit different from the other, but they all have this overall structure. And each one of them must combine with one molecule of beta-2 microglobulin. When we look at MHC class 2, the overall structural shape, especially at the peptide binding groove, is very, very similar. However, there's no beta-2 microglobulin, and the structure is made up of 2 separate MHC genes. So for MHC class 2, we have an alpha and a beta, and we have alpha 1 domain, alpha 2 domain for alpha, and beta 1 and beta 2 domain for beta. And each one of these has its own transmembrane domain and cytoplasmic tail. And these molecules associate with each other in a non-covalent fashion. Now we're going to look at the nature of this interaction between the peptide and MHC. Okay, and hence starting with MHC class 1. There are ways to experimentally strip off peptides presented on the surface of cells by these MHC molecules, and then to sequence them and study those sequences. So here's an example of one sequence, and non-covalent interactions between the peptide and various amino acid residues sitting in the MHC groove. So I don't know how much you remember of amino acids. What amino acid do you think this is? Well, this is clearly tyrosine. Interesting, there's a lot of tyrosines here. That's a tyrosine, and this is a tyrosine. But it doesn't have to be tyrosine. These are two basic amino acids here. So once we strip out these peptides, can we find for any given MHC any common factors? And actually, we do. If you mix together MHC binding grooves and peptides and then see which ones bind and then strip them out and sequence them, we find a pattern. And here is an example for one experiment where 1, 2, 3, 4 peptides have been sequenced that bind well to the MHC groove of our specific MHC molecule. And what we find is a degree, quite a lot of variation overall. Look, Q, F, Q, I, R, P, A, P, arginine, proline, alanine, proline, threonine. Yet when we find two positions near the end, this one near the end, we'll find all these Y's here, and Y is for tyrosine. And it's a coincidence, by the way, that I pointed out many tyrosines. These tyrosines are in the part of the MHC molecule, whereas these tyrosines are actually part of the peptide. At the other end, at the carboxy terminus of the peptide, what's the common factor here? VILI. Well, they're all a bit different, except two I's here. Well, they're all have similar properties: valine isoleucine, leucine isoleucine. These are all bulky hydrophobic amino acids. So there are key anchoring amino acids which are conserved, whereas the rest of the amino acids, there's a huge amount of variability. It doesn't matter too much what they are. That means that any one MHC molecule is able to present many different peptides, which is a very good thing, seeing as we only have in MHC class 1 3 genes, A, B, and C. And if we're homozygous, that's all we've got, meaning that we've got the same polymorphism from our mother and father. However, if we're heterozygous, that means we could have 6. If we could only present 6 types of peptide to T cells, then we wouldn't have a very specific adaptive immune response. However, fortunately, given infection by most infecting organisms, there's a very high chance that we can find within all the proteins in that infecting organism, we can find peptides where we can have a bulky hydrophobic group here and a tyrosine here. Most proteins have a few tyrosines in them, and these proteins get cleaved inside the cell, and we'll look at that starting with the endogenous pathway, how they get cleaved. Now our subject isn't how they get cleaved, but simply just to realize that when the spike protein of SARS coronavirus 2 gets inside a cell, either by infection or with an antigen presenting cell capturing and surveying what's present in its environment, those proteins get broken up into small peptides, and some of them, many of them, may have the necessary conserved sequences at the ends in order to be presented. T cells, and that's our next section and the next lecture, T cells recognize these peptides, but not by themselves. They recognize them in combination with the MHC molecule. So the T cell receptor is actually binding onto the peptide being presented in the MHC molecule. So it has regions which recognize peptide and regions which recognize MHC. So when we come to look at T cell receptors, we'll see that there is as much, or pretty much almost as much, variation in the T cell receptor as there are in the B cell receptor and the antibodies which the plasma cells that make antibodies have as well. So we have this huge variation in T cell receptors, and we have a very limited amount of variation in an individual MHC. In the population, when we look at the genetics, we'll see that there are thousands of different polymorphic forms of MHC. So in the population, we have this huge variation. In the individual, not much variation, 3 to 6 different MHC class 1 molecules. However, each one is able to present many different peptides. Yet, for some pathogens, the MHC polymorphisms that you or I have may not be very good at presenting peptides for a particular organism. So we may have some special weaknesses when it comes to certain pathogens, and we may have some great strengths when it comes to others. Within the population as a whole, we're covered. Some people will have an exquisite, a very high degree of capacity to present peptides from a given pandemic virus, a new pandemic virus. But at the individual level, we'll wait to see what happens whenever some new infecting organism comes, to discover if we have a good set of cards, if you like, this type of analogy with playing cards, a good set of cards or a bad set of cards for that particular pathogen. For MHC class 2, remember class 2, we have more amino acids. It's not just the length, there's also another slight difference between the binding of peptides to the MHC groove of class 2. So here we have class 2. It's a bit longer peptide. Notice that these non-covalent interactions are occurring across the length of the peptide, and also that the peptide is kind of hanging out of the end a little bit. And that's interesting here. So if we do the same type of experiment, look for binding of peptides to MHC and strip out the ones that bind, you can add hundreds or thousands of different peptides, all randomly generated, and mix them together with MHC molecule, and then wash away the unbound ones, and then strip away the ones that were bound. And then again, it's the same experiment as before, sequence what you have. And we find in common with MHC1 that there is a lot of tolerated variation, but also some regions which seem to be conserved. Here we have leucine, phenylalanine, isoleucine. So we've got basically one of 3 amino acids at one point here. And then 3 amino acids after it, we have D and E. And if you remember, these are both acidic amino acids, aspartic acid and glutamic acid. And then 2 amino acids away from that, we have asparagine, lysine, valine, arginine, lysine, 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 arginine, lysine. So there's a bit of variation in the structures of these things, but these seem to be conserved, especially the K, which is lysine, and arginine. What do these have in common? Do you remember from biochemistry? These are both basic amino acids, which are positively charged at neutral pH. The one exception here is valine, which is hydrophobic. I say the one exception. Also, we have asparagine, which does have a nitrogen in its group. However, it's not positively charged. It's hydrophilic. So there's a little bit of variation, but a high degree of conservation here in the structure. Now we move along, another 1, 2, 3 amino acids, and what do we find? In most cases, tyrosine, phenylalanine, isoleucine, methionine, but then we do find other variants here. So the ones I listed are all hydrophobic, but obviously aspartic acid here, that's acidic. Proline is uncharged, it's quite a unique amino acid. And alanine it's hydrophobic as well, but that's quite small. It only has a methyl group. So the conservation is not 100%, but there is a general conservation of patterns at specific positions in the amino acid, in the peptide. But the ends don't all start in the same place. For MHC1, they're all lined up end to end. But here, the beginning is, in this case, 1, 2, 3, 4, 5, 6 amino acids upstream of this conserved isoleucine, whereas in other cases we have only 3 amino acids upstream of the isoleucine. In this example, nothing is downstream of the final isoleucine, whereas for most of the others, many amino acids can be there. So the ends don't really matter very much. because you can have long peptides which just fall over the end of the MHC. For class 1, actually the peptides are constrained, so they must be small enough to fit in here, and you can't have all these long overhanging amino acids. Okay, so now I've given you an overview of the structure of the classical MHC class 1 and MHC class 2. Both of these are molecules that sit on the surface of all nucleated cells, and they present peptides from pathogens. Before talking about the functional differences in MHC class 1 and 2, we've looked at the structural differences of how they interact with peptide, but we need also to talk about the functional implications of that and what effects they have. And basically, to cut a long story short, the difference can be summarized as MHC class 1 is recognized by a co-receptor to the T cell receptor on T cells, which recognizes a molecule called CD8 on the T cell, whereas MHC class 2 is recognized by a different co-receptor on the T cell called CD4. And you may have heard of CD8 positive T cells, which are the cytotoxic T cells, and CD4 positive T cells, which are the T helper cells. So here we have MHC class 2, and this antigenic peptide presented by MHC class 2 would be recognized by a T helper cell. And before we take that subject further, we will look at the genetics of the MHC loci, which in this slide I'm calling the human leukocyte antigen loci. As I mentioned, human leukocyte antigen genes are on chromosome 6, the short arm, and we have MHC class 2, MHC class 3, which I haven't talked about, and MHC class 1. The classical MHC molecules are colored A, B, and C for MHC1. The ordering here is not the same as the lettering, so A and then B and then C. The ordering is a physical ordering, but the naming is based on the history of identification. I suppose if they're all identified together at the same time as we knew their structure on the genome, we would have labeled them differently. But that's the labeling we have. In addition, we have non-classical MHC1 molecules, E, G, and F. If you remember the classical MHC, there's just one chain, and that chain combines with a separate molecule called beta microglobulin. For MHC class 2, we need two chains, alpha and beta. And there are also 3 MHC class 2 pairs, and they're labeled as DR, alpha and beta, DQ, alpha and beta, and DP, alpha and beta. So here in this class 2 region, these are the pairs here, and here we have class 1. And again, we have many other MHC molecules. DO and DM I will talk about briefly later. TAP, I will mention that as well. TAP1 and TAP2. The ones in here I'm not going to mention. I will mention E, G, and F as well later. What about the polymorphism? Well, we keep discovering more and more alleles, and the number is huge. And here is a timeline historically from 1987 coming all the way up to 2023. And along here are the number of these alleles. The light green are the class A and the black is class 2. And look at the numbers here. 26,000 class A alleles. So while any individual is restricted in the number of genes present, the overall degree of variety here in the ability to present antigens is huge within the population. So that's very good at protecting the population from any infectious diseases. Some people may be highly susceptible and may die from some new deadly plague that strikes humanity, but it's likely that many others will survive. The negative consequence of this is if we want to do an organ transplant from a donor to a recipient, then this can only be effective in the case of a compatible match. As you may know, if you know anything about organ transplantation, it's very difficult to get a compatible match. Obviously, we have a huge amount of allelic variation, and it's very rare to have a perfect match. And one tries to have the best match possible, and combining this with drugs which prevent rejection. So all this variation is very bad for organ transplantation because our immune system recognizes HLA molecules which are not self, use the word non-self, self and non-self. So we recognize HLA molecules from someone else as something harmful and reject their tissue. As of January 2024, here are the numbers of alleles that people have identified for A, B, and C. These 26,000 here is putting everything together as just HLA1 or HLA2. If we divide HLA up into the A, B, and C, we've got 8 and 9 and 8,000 different alleles. Now remember that some nucleotide polymorphism may have no effect on proteins, because for any given amino acid, then there can be between 2 and 6 different codons, nucleotide codons, a sequence of 3 different nucleotides which code for that amino acid. So it's possible to have nucleotide substitutions which change the amino acid and others which don't change it. So over here we have a smaller number, which is the number of actual different proteins that are produced. So those can be significant immunologically. Nulls, that means that the result of this polymorphism is is an MHC which is actually not able to bind to any peptides, or maybe even not able to be expressed. So that's a failure. And highly polymorphic, the good news is that all these polymorphisms tend to be inherited together as what we call haplotype blocks. Haplotypes, you may have come across it if you studied human genetics before. And a haplotype block, it's basically a region of a number of alleles which are in what we call linkage disequilibrium. What that means is when you have a chromosome from your mother and a chromosome from your father, and during meiosis, these chromosomes line up together and form tetrads, and then you have recombination happening in order to produce the gametes. And this recombination gives gives a lot of variation, which is why we may look much like our parents in some ways, but not in all ways. So we all have our individuality to that. Obviously, a factor of this is the interaction between the selection of genes on the selected parental, maternal, or paternal chromosomes that we inherit from each parent. So that's one factor. The other factor is this recombination. And the further away any two genes or any two alleles are, the greater the chance of recombination between them. And people have made maps of that and measured that statistically. However, for historical reasons, in our development of human populations through history, sometimes we find alleles which are what we call linkage disequilibrium, which means that we can find two alleles far away from each other, and yet where we find one, we find the other one. So there doesn't seem to be much recombination happening there. That could be because of genetic bottlenecks. So, for example, in recent history, a small number of people from one tribe traveling to inhabit another part of the planet, and then all their offspring have, they're all related from a single common ancestor, which doesn't go back to the beginning of human history. These haplotype blocks are much bigger in Eurasian populations than they are in African populations, and there's a lot of theory about that. I don't want to go too much into the genetics, but that indicates the idea that humans originated in Africa and then at a later point in history migrated to Europe. Okay. Now that's very valuable and important to us. It means that despite all these thousands of different alleles, you don't have to sequence and identify all of these different, all of our genotypes at every single one of these genes, which allele we have. We just need to find a few of them, one or two from each haplotype block. And these haplotype blocks may span a big region, or they may span a much smaller region. But we only need to find one or two alleles in order to basically map our donors and our recipients. The only take-home message I want to give you from here is that in the class 2, sometimes it's a little bit more complicated. On the diagram here, it shows you that there's DR alpha and beta, DQ and DP alpha and beta. But actually, we do have gene families sometimes. So DR, actually, there are many DR genes. There's one alpha, which makes life simple, but there are actually many B genes. So some of us have B1 or B2 or B9, and some of these genes are actually not expressed. Some of them are. We have pseudo genes in here, so it's actually a quite complicated locus or loci. Very dense region in chromosome 6 with many genes on, all of them relating to many aspects of immunology. And the classical ones are expressing MHC class 1 and MHC class 2. And here are some examples of haplotypes for the DR haplotype. So remember, we have blocks where if you have one allele, supposing from here to here is one haplotype, then any given allele in this block is predictive of all the other alleles that you're going to have. And there are multiple blocks. They don't necessarily be end-to-end or the same length. So this slide here is from a paper looking at one of these blocks, which is covering just the DR region here, alpha and beta. And here we have alphas, all the same. Here we have different betas with different allelic forms. And here we have DRB3 and B1. So B1 here and B3 is here. These blue ones are actually pseudogenes. So it's quite complex. We're all very different. We all have well, I say we all have different haplotypes. By definition, we don't all have them, because the definition of a haplotype block is something which is conserved among a portion of the population as a block. So there are a number of haplotype blocks, and for most of us, we fall within any one given of these blocks. However, there are exceptions, and as the number of different alleles being identified increases and seems to be increasing exponentially with time, so we actually find more haplotypes and haplotype blocks. If you ever get involved in molecular classification, you're interested in transplantation and you want to check whether donor and recipient match, then you'd come across a classification scheme. This classification scheme is adopted by the World Health Organization, and it's a molecular classification scheme based upon sequencing. Just to complicate things, there are older classification schemes as well, which you may want to pay attention. You will want to pay attention to if you ever actually practically need to work in this field. But I'm just going to talk about the new version, the WHO molecular classification. HLA, human leukocyte antigen, that's the prefix. And here we have the gene. So this is for MHC class 1, because this is HLA-A. So that's this one over here, HLA-A and MHC class 1. So that's the gene. Then we have a separator, and we have what's called the field group. And the field group is actually the field, which is the first field, is the allele group, also called the serological group. So this is very, very important. People have done serological experiments to identify which variants form within the same serogroup. So we can have some molecular variation, and yet that doesn't result in a serological difference. So you can have many variants, but they're all serologically the same. In this case, 0, 2. Another field separator, and now we come for within that serogroup, we may have a number of molecular polymorphisms, SNPs, which have been identified, and each one is given a number. So we may be interested in that. More field separators, and the next one leads us on to defining whether it's a synonymous or non-synonymous mutation. So a synonymous mutation is a DNA mutation that does not change the amino acid coding. Non-synonymous does change the amino acid coding. And finally, we have a letter here which signals anything to do with expression. So remember that we have null, we may have some polymorphisms which result in no expression or higher or lower expression. So we've got a lot of information here within this classification. Obviously, at this end is the more important one, where we put the gene and the serological group, but you also find these numbers here. So all this polymorphism, where in this MHC molecule is all this polymorphism happening? So here is MHC class 1. We're interested in this, not the beta-2 microglobulin. If we look across the whole of the protein, all of these chains, all of these domains, sorry, alpha 1, alpha 2, alpha 3, we do find hotspots where we find considerable amino acid variability. And then we find other regions where there's very little variation, and then more and less, more and less. Here we've got very little. Why do you think there's very little variation in the alpha 3? None of these peaks are so high. Alpha 3 is down here. The region where the peptide is sitting here, the interface between alpha 1 and alpha 2. So it makes sense that most of the polymorphism which is found is here. That, by the way, is an example of what's called balancing evolution, where there's a selective advantage at the population level, which is the target of, or the unit of evolution, which provides for that population a big advantage in having a lot of variation. It's not the case. There are in many other places where a very specific SNP, a very specific amino acid present at some point, gives a huge advantage to the organism. Others where a specific amino acid may be very harmful, a harmful mutation, and then those positions may be selected for or against. In the case of the MHC, this huge variation within the population is actually beneficial and helping the population as a whole to survive. So that's what they call the balancing selection. By the way, if you find immunology a little bit tricky, it's because it is. First of all, immunology is very complicated because a huge number of different cells, each with their own developmental pathways, work together in a complex system and pattern in order to fight disease. And that means to understand it, we need to understand it in a purposeful or teleological sense. Things are happening for a reason in order to have a final effect, and that's complex. Then there are many biological sciences involved. So now we're looking a little bit at genetics. So we need to understand genetics. Then we're looking at structures, and we need to understand molecular biology. In the slide after this, we're going to start looking at the cell. We need to understand something about the cell biology. So if you find it a bit difficult, well, it's because it is. But I'm doing my best to not make it seem too difficult. Here in red here, with the alpha 1 and alpha 2 domains, are examples of where most of these mutations are happening. So they're all happening at, his perhaps looks like an exception, but most of them are happening at places where the peptide may sit. And for class 2, this data is for class 1, but if we looked at class 2, it's exactly the same pattern. Now, remember that it seems easy now that MHC presents peptides, but that was very strange when it was first identified, and Nobel Prizes were given for this. Studies identified that T cells recognize peptide presented in the context of self-MHC alleles. So this cute cartoon here, we have a dendritic cell, it's presenting peptide, and then we have a CD8 T cell, which is a cytotoxic T cell. He says, sorry, I don't recognize that one. If this T cell comes from another person, then that T cell will recognize probably will recognize the dendritic cell as foreign and start attacking it. But it's not going to recognize this little thing here, which is supposed to represent MHC with a orange peptide on it. So it's not going to see that. It's going to see the whole thing as foreign. So antigen presenting cells presenting actually antigen presenting cells or any cell displaying peptides on the surface of MHC, whether 1 or 2. There's no activation of the T cell unless the T cell and the dendritic cell have the same MHC haplotype, the same alleles. So from this, people realize that the T cell has a dual specificity, means that it has to be seeing the peptide plus the MHC. And in the thymus, we know that that's where actually the T cells which complete their maturation in the thymus, they go through producing T cell receptors, but then in the thymus, there needs to be some further training, and that's called positive selection to make sure that T cells can recognize their own MHC. If they don't recognize, then those T cells are apoptosed and deleted. So all those T cells recognize, all the T cells in your body are able to recognize your own MHC as self. And then any peptides presented on it, you'll find that some of your T cells will be able to recognize those peptides in combination with their MHC. Come and try and get your T cells to see someone else's dendritic cells with peptides. Well, they're just not going to see it. It's quite old work. 1973 was the identification of this for CD4 positive T cells, and a bit later people identified that CD8 positive T cells have this restrictive ability to be activated, and we call that MHC restriction. Okay, now the cell biology, and we're coming near the end now of lecture. We have MHC class 1 and class 2, so I'm starting at the end of the slide if you like, so perhaps easier. MHC class 1 is going to be recognized by CD8 positive T cells, and class 2 by CD4 positive T cells. They're each presenting little peptides from some pathogen, but the way in which these peptides get picked up and associated with MHC is different. And we have these two pathways then called the endogenous pathway for class 1, meaning from inside, and the exogenous pathway for class 2, meaning from outside. So a cell which is infected, say, with Brucella melitensis, and it's going to cause the patient to have humma al-maltiya, in English brucellosis, or infected with SARS coronavirus 2, or any pathogen which gets inside the cell, there's going to be lots of endogenous antigens. That pathogen is going to make lots of copies of itself, and it's going to be full of endogenous antigens, in fact. Well, the cell has this clever way of targeting some of these to the proteasome, and the proteasome is it's not just for immunology, it's also used to get rid of old and unwanted proteins. But in the case of antigens from infecting organisms. The products of the proteasomal cleavage, so the proteasome gets rid of things, so proteins that get sent into the proteasome get cleaved into small peptides. And these small peptides are imported into the endoplasmic reticulum. So this is supposed to represent the endoplasmic reticulum through something called TAP, which is a transports the peptides into the rough endoplasmic reticulum. And this is where MHC molecules, both class 1 and class 2, but we're interested in class 1 now, are waiting. Some of these peptides are going to be a bit too big. Another molecule called ERAP, ER for endoplasmic reticulum aminopeptidase, cuts off some residues from the amino terminus if the peptides are too long. So it trims down the peptides. The proteasome doesn't cut the thing exactly equally into the same length peptides. But ERAP then size checks them and cuts them down to be between about 8 and 10 amino acids long. Then we have what's called the secretory pathway. It's basic cell biology. Secretory vesicles go through the Golgi apparatus and then finally through exocytosis at the cell surface. So the MHC molecules are attached by their cytoplasmic tails to the membrane. They pick up these peptides and then get presented on the surface, so that a cytotoxic T cell can come along and see that and say, aha, this cell is infected, and if we leave this, then this cell is going to end up infecting lots of other cells, so the T cell will destroy this cell. That's the endogenous pathway for class 1. For the exogenous pathway, antigen is coming from outside, and dendritic cells, macrophages, and also B cells present antigens on MHC class 2 in order to get T helper activation, CD4 positive T helper activation. So they sample what's present. In the case of a B cell, then its B cell receptor can sample for things that it's specific for. But for dendritic cells or macrophages, then we need to sample everything. So we have pinocytosis, endocytosis. In various ways, this cell is looking out for anything foreign and trying to pull those things in. And these join the pathway downstream of the Golgi complex. So the MHC is made here, whether 1 or 2. But for the case of the exogenous pathway, then these transport vesicles that go from the endoplasmic reticulum to the Golgi take the MHC class 2 without any peptides. They pass through the Golgi, and here, later, downstream of that, the secretory vesicles join to endocytic vesicles carrying fragments of the exogenous antigen, and this is where they combine. And as you can see, there are two more parts to this story. These two parts relate to the exogenous pathway. Two questions to be dealt with. And the first one is what stops MHC class 2 from being loaded with peptides in the endoplasmic reticulum. So here we've got lots of peptides, and we've got MHC class 1 and 2. MHC class 1 picks up these peptides, but not class 2. Why is that? And the answer to that is in this block, that there's something called the invariant chain, represented by capital letter I and a small I in yellow here, which actually, within the endoplasmic reticulum, binds into that binding groove, thereby leaving it, or thereby it's not available to pick up any peptides. And it's sacrale, it's closed. So the transport vesicle takes this to the Golgi apparatus and packages it as a secretory vesicle, and the MHC binding groove is closed by this invariant chain. The next stage then is taking this closed up MHC1 is how to get the peptides to come in here. Well, what we have now is a new type of endosome called the MHC late endosome, which is a low pH endosome, and in that low pH endosome, the invariant chain is first cleaved, leaving behind a small piece, and the rest of it gets digested. And then that small piece, as this vesicle matures, actually gets released, and it has a low affinity now for the binding groove after this other part of the chain gets digested, and that makes it easy for it to be displaced and for peptide to be put there in its place. So that's how we don't get any peptide binding here, because it's not shown here, but it's shown here. We have this invariant chain which closes up the binding groove until we get to this stage here, which is represented in here. And this picture here introduces some non-classical MHC molecules which were present on the slide. And I did mention that for MHC2 I'll talk about DM and DO. These are regulators that control basically the interaction between this clip here and the MHC, and the dynamics of which one is able to bind more strongly. So we have a DM, which is a positive regulator, which results in release of the clip and binding of peptides, exogenous peptides, to the MHC. And we have a negative regulator as well called DO, which keeps the clip bound in with the MHC class 1 molecule, or sorry, class 2 molecule. So they're all these are class 2. So we haven't said whether this is DR, DP, or DQ. Those are the classical MHC class 2, but it's one of those three. And we also have some of these non-classical MHC molecules, DM and the DO, which are controlling this process. So we can have, through the endogenous or exogenous pathway, MHC1 and 2 expressing peptides, either endogenous or exogenous. Just to make life complicated, we can have something called cross-presentation, and there are cases where exogenous antigens get presented in MHC1 and where endogenous antigens end up being presented in MHC class 2. And this is turning out to be more and more important. Now, the last two slides, we'll just look at how these MHC molecules interact with T cells. So let's take the simpler case first of all. Here's a dendritic cell, antigen presenting cell. So it's going to take up an exogenous antigen. It's searching out for something nasty in the environment, and it's processing this through the exogenous pathway in order to present little peptides on class 2. And here is a T cell, a T helper cell, so that's CD4 positive. Here's a T cell receptor which recognizes the peptide in the MHC groove. We have some co-receptors here, and the relevant one to our discussion today, CD4, which is recognizing MHC class 2. And this dendritic cell is thus activating the T helper cell, but at the same time it's licensing the dendritic cell. So there's a kind of a two-way talk here. The helper cell is getting activated, but this activation process actually is doing something to the dendritic cell, and that's what I'll show you in the next slide. So here we've shaded out the T helper cell. It's there. Here's the dendritic cell, which has picked up the exogenous antigens, presented them here. But this is showing you the cross-presentation. So we've seen now from the last slide that it's very easy. Exogenous pathway, MHC class 2, recognized by CD4 positive T cells, and we've got a T helper cell. That T helper cell is needed for effective activation of B cells. But what about the other T cells, the cytotoxic T cells, the CD8 positive T cells? Well, the T cell helper cell is important there as well. So T helper cells aren't only for B cells, they're also for cytotoxic T cells. First of all, we said that the dendritic cell is licensed now, and it does something funny. It's now presenting exogenous antigens on MHC class 1. So this licensing, sometimes it's called licensing, sometimes it's called activating the dendritic cell. It's the same thing. This dendritic cell now, after activating the CD4 positive T cell via MHC class 2, the T cell sends back a signal telling it, okay, take those exogenous antigens and express them on the surface of MHC class 1. And that's absolutely essential because CD8 positive T cells, they can only see MHC class 1. So how can they get activated? They can only get activated if an antigen presenting cell is presenting antigens on MHC class 1. So that needs this cross presentation where exogenous antigens get presented on the normally endogenous presenter MHC class 1. So here we have CD8 recognizing MHC class 1, again CD3, which we had here, and these other co-receptors. So that's a cross-presenting dendritic cell which has been licensed. In addition, that T helper cell is giving signals, interleukin-2, which is giving a boost to activate further this T cell. Like almost all immunological processes at the cellular and molecular level, more than one molecular signal is required in order for some effector function to happen. So that's the end. I've greatly summarized and reduced the number of slides. So in one lecture, I hope I've given enough of an idea about the major histocompatibility complex, also called the human leukocyte antigens in humans, just a minimum amount of information about the molecular biology of MHC and how antigens are processed and their function, and importantly also the medical relevance in terms of transplantation and susceptibility and protection against diseases at the population and the individual level. The trade-off of having this small number of MHC molecules for each individual against a huge number of different polymorphisms presented in the population is that each one of us is genetically susceptible to various infectious and non-infectious diseases while being highly resistant to others. So on balance, as a population, we can handle all types of diseases, infectious and non-infectious, at the expense that some of us will have a very high susceptibility to diabetes type 1 or some other autoimmune diseases, rheumatoid arthritis. It's a huge list. And for all of these diseases, infectious and non, you can find research papers doing genome-wide analysis and those Manhattan plots that I showed you at the beginning. And the one I showed you at the beginning didn't differentiate which HLA, but also these studies target and identify very specific alleles. Okay, the next and last lecture will be about the T cell. It'll probably be a short lecture. You'll see why when we get to when I give you that lecture. And the final the final assignment will be based on MHC and T cells together, which I hopefully I'll give you the last lecture tomorrow and set also tomorrow the assignment.